Okay, Dr. Jeff Moffitt is an assistant professor in the program in cellular and molecular medicine at Boston Children's and in the Department of Microbiology at Harvard Medical School. Jeff has a long track record of developing in situ single-cell technologies such as MERFISH, and continues very effectively to develop new technologies and new computational tools to answer a range of biological questions from identifying cell types and cell states, spatial organization, all within intact tissues. When I was preparing this, I really enjoyed an amazing review he wrote last year in 2022 on emerging landscape of spatial profiling technologies. I encourage everyone to check it out. And today, he's going to talk to us about genome-scale imaging of the microbiome-host interface with MERFISH. So let's everybody give him a round of applause.
Thank you very much, Stephanie. And thank you very much, Phil, for the invitation to be here, and all of the organizers and Erica for all the hard work to make it possible for us to be here. I'm actually very excited to be here today and to be able to share with you a little bit of some of the work that I've been doing both as a postdoc and as a new faculty member here. Relatively new. It's been a couple years now in my lab. So what I'd like to do today is to tell you about a technology that I helped co-develop, which is an image-based approach to single-cell transcriptomics. It's a technique that's called multiplexed error-robust fluorescence in situ hybridization. That's a mouthful, so we're going to call it MERFISH throughout the talk. And in a nutshell, it's an approach that allows us to image and identify hundreds, thousands, or even more individual RNA molecules within intact single cells within intact tissue slices. So that allows us, in turn, to create molecular and cellularly defined atlases of a wide range of tissues. What I think is exciting about this technology is that it not only gives us the ability to unlock new windows onto a really broad range of biological questions, it also raises a tremendous number of new and exciting computational challenges. So what I'm going to do today, I'm going to start with an introduction to MERFISH, and then I'll tell you two biological stories from my laboratory focused on a system that we're very passionate about, the micro-host interface. And I will highlight through those biological stories some of the computational challenges that emerge with these type of data, and perhaps some of the simplistic ways we started to address those, hopefully to inspire some of you to see if you can help us develop even more sophisticated ways to extract biological meaning from these data. Before I go further, though, I just want to acknowledge that I'm a co-founder and a stakeholder in a company known as VisGen. It is commercializing MERFISH, though none of VisGen's products or services were used to generate anything that I'll show you here today. So let's see if I can. There we go. So I thought I would start by giving you perhaps a high-level motivation for why one might want a spatially resolved approach to single-cell transcriptomics like MERFISH. And I thought I'd do so by sharing with you a story from when I was young. So when I was young, I used to like to do a lot of auto mechanics with my father. And so with that in mind, I hope you can understand why when I went out one day to our 1987 Ford Ranger and turned the key in the crank and this horrible white smoke poured out the tailpipe, why I was so absolutely thrilled. Because if you have white smoke, that means you're burning oil. And there's only one way you're burning oil, which means you've cracked the head gasket. And there is only one way to fix that, to pull the engine out of the truck and completely disassemble it. That's exactly what my father and I did. We had the engine completely disassembled on our garage floor, and I had this sense when I could see all the parts of this complex machine in their glory that I truly understood how this system worked. Of course, that was a very fleeting sense because my father said, okay, now that we fixed the gasket, can you put it back together? And of course, I didn't remember how these parts went together. So the point I'm trying to make here is that one of the most powerful ways to understand complex systems is to disassemble them into their individual parts. You can catalog those parts and understand their individual functionality. But to truly understand the system, you need to know how those parts fit together. And I think this is an apt analogy because we really are living in the era of biological disassembly. Techniques like single-cell RNA sequencing, which I know everyone here is very familiar with, allow us to essentially take any complex biological system tissue, dissociate it into its individual parts, cells, capture individual cells, extract their RNAs, and sequence them with next-gen sequencing. And with these molecular profiles defined at the transcriptome scale, we can do some really remarkable analyses. Discover and define de novo cell types and cell states. Because we're defining these individual cells based on the genes that give rise to proteins that give them function, we can actually we can infer quite a bit about their functional behaviors as well. So these are the tools that allow us to catalog parts. But as I think you fully appreciate, tissues are not amorphous collections of cells, and cells are not amorphous collections of RNAs. Tissues are intimately organized spatially, and this organization is a critical determinant of how cells communicate with one another and how the behaviors of individual cells give rise to behaviors of the tissue as a whole. And on a smaller scale, as you can see here on the right, the RNAs within cells themselves are spatially organized. And it turns out that this internal organization, the transcriptome, is particularly commonplace in eukaryotic systems and can play a very powerful form of post-transcription regulation. So the point I want to make here is that having a list of the RNAs that are found within a cell and a catalog of the cell types found in a tissue, while powerful, leaves many biological questions unanswerable. So what we need are technologies that can merge the strength of single-cell transcriptomics while preserving this intracellular and tissue-scale spatial resolution. That's what we set out to build when I was a postdoc, this technology that we call MERFISH. But it's actually been an exceedingly exciting time, as I think many of you are aware, in the field of spatial transcriptomics over the past, say, 7 years or so. And there's been an explosion of technologies that can give us genome-scale gene expression information with preserving elements of spatial context. I won't say much about this landscape, other than it's very exciting to see a huge amount of diverse and creative technologies here, but it can be broadly organized into spatial capture methods that essentially imprint spatial information on short barcodes attached to RNA molecules that can then be identified via sequencing, or image-based methods, methods that generate the identity of individual RNAs by creating what I call optical barcodes. There are a huge number of methods in both of these categories, and not just academic methods, but commercial methods, as you see here in red. And these two different suites of technologies have very nice complementary strengths and weaknesses that I'd be happy to discuss in questions. I'm going to talk about image-based approaches. MERFISH clearly falls. And then before I get into the details of MERFISH, I thought I would share with you a little bit of what gets us excited in my laboratory. This is an image that inspires us. This is a cross-section of the adult mouse colon. In red is the sea of bacteria that comprise the microbiome. In green is the mucus layer that separates those bacteria from the host. In purple, you see the nuclei of the diverse host cells in the host gut. And we are fascinated by questions associated with the spatial organization in this system. We're asking questions associated with how organization shapes interactions within the microbiome and keeps this remarkably complex community stable. We're interested in how spatial organization and the diverse cell types that are found on the host side shape interactions with microbial communities. And we're very interested in what happens when this barrier between host and microbe breaks down. Why we find this image so inspiring is not just because I think you can see how intricately organized this interface is at the cellular scale, but because what this image doesn't show. Hidden in the sea of red are hundreds of different bacterial species expressing hundreds of thousands of genes. And even on the host side, the same genome gives rise to perhaps 100 different cell types and states. Each of these plays a differential role in the dynamics of this complex interface. And so the challenge here is the sheer complexity of this system. And we're solving this with genome-scale imaging tools that leverage this basic concept in spatial transcriptomics. So today I'm going to start by giving you an introduction to MERFISH. I'll tell you the basic concepts behind this technique, how it works, how it performs, and I'll highlight some of the exciting low-level computational challenges that we face. Then I'm going to tell you 2 stories. The first is going to focus on what happens in the mouse gut when this barrier between host and microbe breaks down. And I'll show how we can use MERFISH to chart both the cellular and spatial remodeling in the context of gut inflammation. And then I'm going to tell you an unpublished story that we're just extremely excited about in my lab, where we've now been able to move MERFISH from the mammalian host side into the microbes themselves. And I'll show you how that can allow us to profile how bacteria fine-tune their gene expression based on the different niches that they can find themselves in the gut. MERFISH is based upon a very powerful technique known as single-molecule fluorescence in situ hybridization. In this approach, you fix your sample, permeabilize the membranes of your cells, and hybridize to it a set of fluorescently labeled DNA oligonucleotide probes. These probes bind multiple locations on the RNA of interest, and that concentrates a large number of fluorophores, so that if you then image the sample with an epifluorescence microscope, you get a bright fluorescent spot for each molecular copy of that RNA. So now if you want to determine single cell expression level, you just count the number of spots per cell. Now what we wanted to do in developing MERFISH was to take this very powerful technique and push it to the transcriptome scale by multiplexing on a massive scale. We do that conceptually by assigning to different RNAs individual binary barcodes. And then we read those barcodes out in the following fashion. We stain our sample using fairly conventional single molecule FISH approaches, but instead of targeting all RNAs that we're interested in, we're only targeting the RNAs that have a 1 in the first bit assigned to them. We get a pattern of fluorescence, we image the sample, remove the fluorescence, and then restain exactly the same sample It's targeting every RNA that has a 1 in the second bit. We get a different pattern of fluorescence, we image, extinguish, and continue. So we have 1 round of single molecule FISH for each bit in our barcode. And the fluorescent on-off pattern we see for each molecule serves as an optical representation of that barcode that allows us to not only tell you where individual molecules are found, but their identity as well. This ability to build optical barcodes through sequential rounds single molecule FISH is the first critical concept that underlies MERFISH. The second concept, which I won't say much about, but is actually critical to high performance in this technique, is that we borrow ideas developed in the context of binary communication to handle readout errors. So you can think about if we make a measurement error in one of these single molecule FISH rounds, we could be flipping or corrupting these bits. Turns out this is a problem that has had solutions for decades. We borrow these error correcting and robust encoding schemes. We use those to encode our RNAs, and that gives us very high performance here. That's the in MERFISH. So let's take a look at some actual data. This is a MERFISH measurement of 140 genes in a set of human osteosarcoma cells. You can see 3 cells there. This is after the first round of staining. Each of those bright little spots is a single RNA molecule. If you zoom in on one region in one of these cells, you can see one of these molecules, and its presence in this image gives us a 1 in the first bit. We remove this signal, restain the sample. That RNA is no longer fluorescent. That gives us a 0 in the second bit. Repeat this process. The RNA is back. It's 1 in the third bit. And I think at this point you get the idea. We're going to do this 13 more times to build up a stack of 16 images. That RNA is fluorescent in the following 4 rounds. That gives us the binary barcode you see at the top, and it allows us to identify that RNA. Of course, we have a barcode like this for each of the different RNAs we targeted, which allows us to reconstruct an image like this. Each marker is a single molecule, and the color is the identity of that molecule. Great question, Rafael. So what you see here, the pixel is smaller than the optical point spread function of the microscope. So the fluorescence from a single molecule is spreading across multiple pixels. And I won't say anything more here, but being able to translate these on and off patterns of fluorescence into decoded RNAs is actually a particularly difficult computational task, even though it seems perhaps conceptually simple. And we've worked quite a bit on that. I'd be happy to answer any questions if people are interested. So let's now ask how well MERFISH performs. So what we did is we made these measurements of 140 genes across thousands of cells. We computed the average for each gene across those cells, and then we compared to an independent technique for determining RNA abundance. That's bulk RNA sequencing. That's what you see here on the left. Each dot is a MERFISH measured gene, MERFISH on the y-axis, bulk sequencing on the x. I think you can see that these 2 very different techniques for determining RNA abundance actually produce measurements in high degree of correspondence. This gives us the ability to know that MERFISH measures relative RNA abundance quite accurately. But to determine absolute abundance, we turn to the gold standard for single molecule expression measurements, which is conventional 1 at a time single molecule FISH. We did that for 15 different genes, and that produced the scatter plot that you see here. And I hope you can see that, again, these 2 different techniques agree very nicely. But there's several things that I want you to notice about this plot. The first is that MERFISH produces measurements that agree with single molecule FISH across the full dynamic range probed here, over 3 orders of magnitude. It produces measurements that agree all the way down to the lowest expression level we probed here. That's single molecule sensitivity. And these markers fall along the dash quality line, which tells you that if there were 100 copies of an RNA in the cell, MERFISH would count almost all 100 of them. And if you think this is actually, if you stop to think about it, this is actually pretty remarkable, what I've just told you. We can use this technology to increase the multiplexing of single molecule FISH by at least 2 orders of magnitude with basically no cost and performance. With the same dynamic range, same low sensitivity, and same high detection efficiency. And so, of course, that inspired us and others to think about how high you can push this multiplexing. The samples that we produce are basically indefinitely stable. So we can have as many bits as we want. So the number of rounds of imaging was never the limitation to our ability to define numbers of RNAs. What is the actual limitation is the density of the molecules. As you start to target more and more molecules, the fluorescent signal for molecules starts to overlap. And as soon as that overlaps, you can't make sense of these barcodes. And you can see an example there on the left. This is, again, a human osteosarcoma cell. And here we've stained very many very abundant RNAs. And I hope you can see there's no resolution of single molecule signals. So there are multiple ways to solve this problem, but they all come down to the same thing. We have to dilute these signals. And the way we often do this is we use a technology called expansion microscopy, where we physically swell the cells. Now these individual signals are separate. We can resolve them. And this was work done by my postdoctoral mentor, where in her lab they showed that you can now resolve 10,000 different RNAs with the same high performance that I just showed you for 140. So if you can address this RNA density level, you can tune the multiplex in this technique anywhere from hundreds of genes all the way up to near transcriptome scale. With this ability to perform massively multiplex single molecule RNA imaging measurements, you can actually start to tackle pretty wide and exciting range of biological questions. And I'll just give you a very brief survey. For example, because your detection efficiency is so high, the natural variation you see in gene expression from cell to cell reflects true biological variability, not the Poisson noise of whether or not you detected that molecule. So now you can look for covariation in gene expression from cell to cell, and you can use that to back extract properties of regulatory networks, because noise in those networks propagate through the network and produce correlated gene expression. Of course, you don't just know how many copies of an RNA are found in each cell, you know exactly where each copy is found, so you can map the internal organization of the transcriptome. And you can even use that information to infer properties of cell state. RNAs in eukaryotes are born in the nucleus and often degraded in the cytoplasm, so the ratio of nuclear to cytoplasmic counts can tell you something like an instantaneous transcriptional rate or an RNA-like velocity. And what I'll focus on mainly today is that we can also use these measurements in intact slices of tissues to define and discover cell types, all while mapping their organization. And you can also do some really cool biotechnology applications where you use synthetic RNAs to work with different genetic perturbations, though I won't say anything more about that. So that brings us to the first vignette that I want to tell you about. I'm going to tell you about my lab's first major effort to use MERFISH in our own independent work. And this was focusing on aspects of inflammation in the mouse gut. So I think many of you are aware that inflammatory bowel disorders are on the rise in the West. And these are diseases that are typified by chronic and relapsing inflammation. One of the major challenges to understanding the origins of IBD is that many different cells contribute to IBD. And these cells are intricately organized in the gut, and that organization changes throughout disease, which shapes the ways in which these cells can interact. And so we reasoned that a technology like MERFISH, which can both define cell type and organization, might be able to provide some new insights into inflammatory bowel disorders. And so we partnered with Roni Narwarski's lab to use a common mouse model of IBD, which I'll get to here in a second. But the first thing that we had to solve in my lab was how we could generate high-quality MERFISH images in the gut. So let's take a look at what, this is the most important thing I say here. The work I'm about to show you, it was done by this talented team of postdocs, Paulo, who's in our laboratory, and Keisha, who's in Roni's, supported by these very talented graduate students in my lab, Rosalind and Evan. So this is what a cross-section of the mouse colon looks like in a healthy colon with MERFISH. Each tiny little dot is a single RNA, and here we've only colored 8 different RNAs to get you a sense of the structure of this tissue. To give you a deeper sense of the richness of this tissue, let's zoom in on that black box. And here in this animation, we're going to go through a series of different RNAs. So each little gray dot is the one of roughly 1,000 different RNAs we measured in this experiment. And as the animation plays through, each blue dot is each instance of the RNAs listed in the top. So these images can provide a first-order validation of the accuracy of our measurements. For example, we're coming up here on a pan-epithelial marker. You can see that defines this epithelial layer in the gut. These are markers of epithelial development all the way down to the stem cell niche. These are markers of different fibroblast populations found in the lamina propria. Those are muscle markers found in the muscularis externa. Those are even markers of the myenteric plexus, a portion of the enteric nervous system. So what was exciting to us is all these markers were found exactly where you'd expect to find them. But we can do the same type of analysis that I showed you in cell culture, where we can average expression across individual slices, determined via MERFISH and via bulk RNA sequencing, and see to what degree these technologies are in concordance. And here in the scatter plot, MERFISH on the x-axis or y-axis and bulk sequencing on the x, you can again see that these approaches agree very nicely. I wanted to show this for another reason, though. I want to highlight something that is common amongst all image analysis or image-based transcriptomics approach, which is that at low levels of expression, you see we lose this correlation with bulk sequencing. So while this technique has many similarities to single-cell sequencing, that loss in correlation represents a finite but real false positive rate. There are RNAs that aren't there that we do count, but that false positive rate is extremely low, less than 1 count per 30 cells. Nonetheless, I think it's important to keep in mind because the different image-based technologies that are now emerging can have very different performance in these false positive rates. And that can have a profound consequence on the type of biological inferences you can make from your data. So being able to define these RNAs in space was only the first challenge that we faced. To be able to define cell types and states, we need to partition these RNAs into individual cells, and we need to be able to look in our slices and determine where one cell ends and the next begins. I think you're all familiar with this problem. This is known as the image segmentation problem. I want to walk you through several of the approaches that we have taken to addressing this challenge. This is by far not a solved problem, and this is another place where there can be a tremendous amount of further innovation and computational methods for these type of measurements. First thing we do is we incorporate immunofluorescence into our measurements by oligotagging our antibodies. That way, the same repetitive fish staining that we use to read out RNA barcodes can be used to read out the presence or absence of antibodies. And then we use a pan cell surface antibody against, an antibody against a pan cell surface marker to provide the boundaries of individual cells. This is a cross-section of the mouse ileum stained with an antibody to the sodium potassium ATPase that's found on the surface of every polarized cell. I hope you can see by zooming in that this is a very information-rich stain, and we can see many of the boundaries here. And then we use basically some very well-defined machine learning algorithms. This is CellPose that we've also used at Mesmer as well. We can train on these images, and then we can pretty robustly define cell boundaries. But I want you to notice here that even though this has done a very good job, there are plenty of places where cells are missing, either because we haven't trained enough or the costain's just not clear. So we knew that we needed things that would allow us to supplement this type of segmentation. So fortunately for us, Peter Karchenko's lab was working on this, and we partnered with them to test out a really very brilliant idea they had. Their notion was that we could use the distribution of RNAs themselves to help guide this segmentation problem. And here the concept is simple. If I have two different cell types, like a B cell and a T cell that are physically touching, if I look at the physical distribution of B cell and T cell markers, as I pass from one cell to the next, I should see the B cell markers go away and the T cell markers come up. So the physical distribution of these RNAs can guide the segmentation of images. This is the MERFISH measurement we did across that cross-section of the ileum. Here, to test that idea, we're going to color RNAs not by their identity, but by the composition of RNAs around them. That's what you see here. And if this information of RNA distribution is sufficient to help guide cell boundary decisions, we should find that when two different cell types touch, we see a sharp change in the color. There's a lot going on the left. All I want you to do is to look at that bottom right corner, and I hope you can see that indeed. When you have two different cell types touch, you go from blue to brown. So Peter's lab built an algorithm called Baser that exploits that information. They can partition these RNAs into individual cells. Now RNAs are colored by the cells to which they've been assigned. And then we can overlay that on the immunofluorescence staining to see how well it's done. I still find this remarkable. Baser, with no information of the underlying distribution of these cells, nonetheless does a remarkable job of assigning RNAs to cells in a way that's consistent with these boundary markers. It's not always perfect. You can find plenty of examples where this fails, especially when two cells of the same type touch. And so what we routinely do is use CellPose with these immunofluorescent boundaries to provide prior probabilities that Baser then can incorporate as it makes this decision on how to partition these RNAs. And we've been very excited to see that this approach of combining machine learning with an RNA-based method like Baser has really become a standard in the field. Okay, so now we can partition RNAs into cells. And so we can now define cell types and states and map their organization. Let's turn to this mouse model of inflammation. This is a classic chemical-induced colitis model. You add this detergent, dextrose sodium sulfate, to the drinking water of the mouse. You do so for 7 days. Over that period of time, the mouse can lose something like 30% of its body weight. And then we retract the DSS from the water, and the mouse over the next 2 weeks will recover its weight. We're going to sample mice before the treatment, early in the treatment, at peak weight loss, and at recovery. And then we're going to use MERFISH to profile about 1,000 genes. So when we think about designing panels, we don't think about just leveraging, say, single-cell data to allow us to register cell types in space. We also want to try and define something about the functionality of these cells as well. And so the way we designed this panel is actually a pattern that we've repeated across many projects. The first part of the panel is classic cell type markers that allow us to define what individual cells actually are. But the rest of the panel are target pathways that are relevant to the biology that we're interested in. For example, in inflammation, cytokines and cytokine receptors are a natural category of genes that play a big role in how cells polarize and are recruited. And so what we do is we include all of these genes, even if they have no evidence of expression. And that's what's comprised this panel. So now let's take a look at what we get when we run MERFISH in this mouse model. So in total, Paulo imaged something like 20 mice, 2 different slices, and generated a data set comprised of about 1.3 million cells. What you see on the left here is a UMAP representation of these cells, colored by the major classes of cells. It's common in this field to use single-cell data to help interpret this, or to project single-cell data into space. But when you have the sensitivity to get with MERFISH, you don't need to do so. So everything I'm going to show you from now on out is UMAPs that are analyzed just from MERFISH data alone. And so it was very satisfying to us that we could see the major divisions of cell classes you would expect in these data. But of course, we also know exactly where they're found. And these are the 52 different slices that Paulo imaged. We can color each of these cells based on the major class of cells they've been assigned to. So you can see epithelial layers, muscle layers, immune recruitment throughout disease, and so forth. I also want to highlight that this is a very image-intensive technique. So what you're seeing here represents about 50 terabytes of raw image data. It's actually a relatively small data set for us now. We just built our 2nd petabyte of storage in my lab, which we manage ourselves because we can't afford to do it any other way. And that, unfortunately, is filling up rapidly. So this is the future of a lot of spatial biology. But we're not just able to define epithelial cells from immune cells and so forth. We can actually take those individual populations and subcluster them. And we were struck by the remarkable diversity we were able to define with MERFISH. So what you see here in the upper left is a UMAP representation of just our epithelial cells. If you look at the classic genes, which I'm not showing you, we see the full diversity of epithelial populations you'd expect to see. Stem cells, transit amplifying cells, mature colonocytes. We see M cells, we see tough cells, we see goblet cells, and so forth. What you see here on the right are 4 different example slices. Gray marks the center of each cell, and the colored cells are epithelial cells colored in the same way that they are on the UMAP. These 4 different slices are taken from different stages in disease. So you can see a healthy mouse in the upper left, and as we proceed through inflammation, I hope you can see that there's profound changes in the epithelial cell type diversity and organization across this mouse. But of course, in this measurement, we don't just see epithelial cells, we see the same rich diversity in fibroblasts, in immune populations, in endothelial populations, in populations of the enteric nervous system, and even the muscle layers. There are actually also a few stray adipocytes that we captured, as well as multiple populations of interstitial cells called pacemakers in between the muscle cells. And that's one of the things that excites us about these type of data, because you basically see the full cell diversity. Even if you're interested in one compartment, you see everything. There's a lot we've learned about the cellular polarization that happens in disease, but I just want to gloss over that so I can get into a few more interesting things in terms of computational analysis. The most important thing is that we see that many different cell populations actually polarize in inflammation. This is an epithelial UMAP. The arrows highlight ways in which healthy populations polarize in disease. We call those polarized states inflammation-associated epithelia, or IAEs. We see the same in fibroblasts, and the same in muscle cells. We actually see it in all cell compartments, that inflammation induces gene expression changes. Now, that allows us to define the cellular remodeling that happens in disease. What we were far more interested in is in the spatial remodeling. And here we faced a challenge that I think many of you are aware of. How do you statistically define recurrent spatial patterns? And how do you quantify the remodeling that happens in a tissue? So one way that people will do this is they will define a common coordinate system. In the brain, for example, this is very common. You define specific cortical layers, for example. And as you induce disease, you can ask, how does cell type abundances change in those specific compartments? What you see here on the left, the first slice on the very left is a healthy mouse where cells are colored by cell type, and on the right is a diseased mouse. In healthy mice, there is recurrent spatial patterns, and perhaps we could have taken such an analysis. But what I hope you can see is that in disease, the regularity you see in the mucosa is basically gone. There is no common coordinate system to reference. So we needed to find a coordinate-free way to represent spatial organization. And so we developed an approach that actually mirrors what others developed independently. We take for every cell and we quantify the number of different cell types in its immediate neighborhood. That gives us a vector, which we call the neighborhood composition vector. We can treat that vector just like you would treat a vector of gene expression in a cell. We can project it from its high dimensional manifold space into 2 dimensions with UMAP, and we can identify statistically recurrent collections of cells, what we call cellular neighborhoods, using lighting clustering. That's what produces the UMAP you see here, and this is a statistical representation of spatial organization of tissue. If you were to look at the different abundance of cell types in different corners, you can see that different corners of this UMAP are represented by different cell types. And what was very satisfying to us is that if we then take those individual cell neighborhood labels and color cells based on the neighborhoods to which they're assigned rather than their type, the anatomical organization of tissue basically falls out for free. That's what you see here on the right. Let's dig a little bit more into these neighborhoods. And what you see here is a UMAP representation of epithelial fibroblasts and immune cells, colored not by their identity, but the neighborhood to which they've been assigned. And on the right, I have one healthy slice of the mouse gut, where I've colored the 3 neighborhoods that define the mucosal layer. The blue neighborhood is the bottom, where the stem cells are found. The green is the intermediate in the mucosa, and the gold is the top. What I want you to show is that these neighborhoods not only capture changes in cell type composition, they actually capture subtle variations in gene expression that you can see in the UMAP. And you're going to see that because as I walk from the bottom of the crypt to the top, I want you to see across this UMAP that we're going to continuously transition from the bottom of that corner of the epithelial UMAP to the top, and that specific corner of the fibroblasts at the top, as well as even immune populations like macrophages. So you can see as I walk from the bottom to the top, I hope you can see there are continuous variations in the UMAPs that are defined by gene expression. So what it tells you is that neighborhood occupancy can capture subtle variations in gene expression. Again, I think that's an exceedingly interesting computational challenge for these type of data. This is perhaps the simplest way to look at this type of information. Let me say a little bit of what we see in inflammation, and I'll move quickly here to highlight some other aspects that I think perhaps may be more interesting. What you see here is a slice on day 0, healthy mouse, and early in inflammation. Colored are the specific cellular neighborhoods associated with the mucosa. I just walked you through what we see in healthy. What I want you to see is that early in inflammation, the gut basically looks histologically completely normal. But on a molecular and cellular level, it is completely changed. We lose the stem cell neighborhood, and we see the emergence of this new inflammatory associated neighborhood called mu4. This underneath is defined by 3 of these different inflammation-associated populations that I described. There's a lot we've learned about the biology of this process from the genes they express. All I want you to take from this, which I think is really absolutely beautiful, is that even though the gut is exposed to this small molecule that is uniformly filling the gut, the response is remarkably spatially patchy. You can see that in the distribution of these cells, and you can actually see it very clearly in the distribution of the genes that define them, particularly these 2 genes. I hope you can see that even though the gut sees exactly the same concentration of the dextrin sulfate, coherent regions of gut polarize together. I think that speaks tremendously to the mechanisms that reinforce inflammation-associated polarization in local neighborhoods. That type of spatially patchy response we see continues on as we proceed into much more severe inflammation. And what you see here on the left are 2 slices taken from peak weight loss. This is day 9. Blue marks otherwise healthy neighborhoods, and green, red, and pink mark severely inflamed neighborhoods. These are 2 slices taken from different mice, and I want you to see that different mice look very different. Even slices taken from the same mouse look very different. Nonetheless, the boundaries between these neighborhoods are remarkably sharp. You go from this severely inflamed neighborhood in red to this less inflamed neighborhood in green over the distance of microns. It turns out that's not an artifact of our analysis, but a reflection of the sharpness of the distribution of these cell types to define those neighborhoods. So here what I'm plotting are example cell types that define this red neighborhood, and you can see remarkably sharp boundaries when red goes to green or to blue. That's also reflected in the underlying genes as well, which I'll just show you here. I think you get the point. What's interesting about these red neighborhoods is they represent ulcers. They're devoid of epithelia. Presumably, this is a place where the microbiome can impregnate into the tissue and trigger a variety of immune responses. We see they're also defined by the polarization of a series of fibroblasts and the recruitment of a series of very interesting novel immune populations. Macrophages are monocytes that are producing large quantities of IL-1 beta, as well as not 1, but 2 different populations of neutrophils. What's also very powerful about these methods is that not only can we define different spatial neighborhoods, we can actually capture very subtle variations in gene expression based on the location of cells with respect to anatomical features. And that's particularly true for some of these activated fibroblasts in the ulcer. What you see here is an ulcerated region, and I'm plotting the distribution of 4 of our activated fibroblasts. You can see that those fibroblasts are not uniformly distributed through the ulcer. They're structured differently, with this IAF4 found right at the luminal surface. But what was very exciting to us is we noticed that in the gene expression UMAP for IAF4, there were gradients in gene expression that you can see in these 4 UMAPs on the right. You can see there's a concentration of these genes. And if we look at where those genes are expressed, they're actually found exactly at the luminal surface. This IAF4 has gene expression gradients that are set based on its distance to the surface of the lumen. And what's exciting about these measurements, of course, is that we can detect these very subtle and potentially functional differences. I also want to highlight something that we noticed on the immune population that also, I think, reveals some of the promise of MERFISH-like measurements. So we noticed, as I mentioned, not 1, but 2 different types of neutrophils. And what you see here in green and red is the UMAP portion of our immune UMAP that represents these neutrophils, and you can see that we've clustered them into 2 groups. But in reality, there's plenty of neutrophils that fall right in the middle. So we did the classic type of analysis. We did a pseudotime analysis, and we found that you could find continuous gene expression gradients from neutrophil 1 to neutrophil 2, and we could find the genes that define those gradients. What's exciting is the genes that are found in the middle are all AP1 transcription factors that are often induced when you are transcriptionally remodeling. And what we show is that this subtle variation in gene expression actually correlates to the structure of the ulcer. Neutrophil 2s are found predominantly outside the ulcer. Neutrophil 1s are found inside the ulcer. And what we believe we're capturing here is the gene expression program that directs these neutrophils to be excreted out into the lumen. So now we've been able to define some features of this ulcer structure. One of the other things that we can do, like other single cell methods, is we can perform receptor ligand interactions. But of course, the advantage we have is that we can spatially prioritize those interactions because we know that these cells are physically adjacent. For any of you who have done receptor-ligand interactions with beautiful programs like CellComP or NicheNet or CellChat, the problem is not finding hits. The problem is sifting through the thousands and thousands of hits that you get here. So being able to spatially prioritize those hits, I think, is very powerful. But even when we spatially prioritize the potential interactions that we see in this ulcerated neighborhood, there's a mess of biology here. So let's just examine one potential mode of interaction. Let's ask what recruits neutrophils to this ulcer. So these neutrophils express CCR1, the chemokine receptor CCR1. You can see that other immune populations, Tregs, dendritic cells, macrophages, are expressing the cognate ligands and are helping to recruit these neutrophils. They also express the chemokine receptor CXCR2. What I want to highlight here is that the fibroblasts, the IAFs that you see there, are the predominant source of these chemokines. And so activated fibroblasts are playing a predominant role in recruiting neutrophils here to the ulcer. I also want to note that these neutrophil 2s that are out in the lumen are also recruiting those neutrophils into the ulcer. And what's remarkable from our pseudotime analysis is that once they're excreted in the lumen, they turn down CXCR2, presumably so they're not recruited back into the ulcer. So these are the type of cartoon representations of the biology that we can learn from this analysis. I just want to highlight that what we saw in the mouse model actually has some potential implications for human disease as well. Inflammatory bowel disorders like ulcerative colitis or Crohn's disease not only are on the rise, but about half of all patients eventually develop disease that's resistant to our standard of care. That's an antibody against tumor necrosis factor alpha. And so many labs have looked for biomarkers that can explain or predict whether a patient will be responsive or non-responsive to this treatment. And what you see here on the left are the mouse homologs of a panel of human biomarkers derived by Fiona Power's lab. And so we wanted to know, are these biomarkers that predict non-responsive to tumor necrosis factor alpha enriched in any of our specific cell types or neighborhoods? And the color here indicates the enrichment of these in specific neighborhoods. The neighborhood U8 is our ulcerated neighborhood. And that was really remarkable to us because we find that if we look at the individual UMAPs, almost all of these human biomarkers, their mouse homologs, are selectively expressed in different populations of cells in this ulcer. And so if you permit me to be a bit provocative, what this tells us not only is that mouse can help us inform human disease, it tells us that the true biomarker of disease is ulceration. That's what determines whether or not you are going to be responsive to tumor necrosis factor alpha. And this is a very reassuring thing because the histopathologists have been telling us this for 10 years. And so I want to illustrate that this is one of the strengths of these spatial transcriptomic methods, is it really can merge these distinct molecular and cellular views with spatial views like histopathology. I suspect I have very little time to discuss our bacterial work, but I'm gonna do the bad PI move of trying to see how far I can get into this. What I'll say is that we've been really passionate about trying to understand gene expression on both the microbe side and the host side. Thank you. One of the central challenges that we face in doing MERFISH in bacteria is their remarkably small size. So bacteria are perhaps 2 microns in dimension. The optical point spread function of a microscope is not that much smaller than that. But what's really problematic is even though bacterial cells are small, their RNA copy numbers are not that small. So bacterial RNA densities are 3 orders of magnitude higher than eukaryotic RNA densities. It's remarkable when you think about the difference in the cytoplasm between these 2 domains of life. And so we knew we had to solve that problem. Somehow we had to span 3 orders of magnitude in RNA density. And so we do this by using a technology invented at MIT by Ed Boyden's lab called expansion microscopy that I mentioned briefly before. We anchor RNAs of interest to a charged hydrogel. We digest everything else, remove the salt that screens those charges, gel physically swells, and it separates our molecules apart. This is work done by the very talented postdocs and graduate students that you see in my lab here, Yanyu, Ari, and Nana. And what I want to show you here is that we now have a toolbox of tunable expansion that allows us to go from regular E. coli cell, which is perhaps a few microns long, that's what you see here, E. coli stained with the 16S ribosomal RNA stay, up to 100-fold expansion in volume, all the way up to 1,000-fold expansion in volume. So these are now bacterial cells that are bigger than many eukaryotic cells. This allows us to span these 3 orders of magnitude, and now we can do MERFISH in bacteria. Individual RNAs here, again, are marked by markers, and the color is their identity. Here we're profiling half of the E. coli transcriptome in a single measure. This in turn allows us to do a lot of exciting questions. For example, we can start to ask about cellular heterogeneity in bacteria. So we did the classic Monod experiment where we take bacteria and shift them from 2 different carbon sources, and we asked if the population responds as a whole. This is a UMAP representation of E. coli as it proceeds to starve itself of glucose and then start eating xylose. And what I want you to see is there's a tremendous amount of cellular heterogeneity that we find there. I won't go into the details of the genes here, but what we find is that bacteria in different stages, not only in different stages of this growth curve, are different, but there are distinguishable subpopulations of bacteria responding differently to starvation in each of these populations. Another thing that was really striking to us is that not only can we look at differences in gene expression from bacteria and capture heterogeneity, we can also chart the intracellular organization of the transcriptome. These bacteria are a couple microns long. If I let go of a molecule, diffusion mixes that in the cytoplasm in a matter of a minute. And yet, the bacterial transcriptome is intricately organized. Here are the average distribution of 3 different genes. You can see genes that are found in the cytoplasm, genes that are found in the cell wall, and genes that are found at each of the poles. We can do this now for essentially the whole transcriptome, and we find that there's a diversity of cellular organization patterns that we see here. Functionally, we have no idea what this means, but we're very excited about the fact that we can measure it. And finally, we can get to the experiment that we built all of these tools to do. We can now ask if bacteria in the complex environment of the gut can change their gene expression in response. So we took the common human commensal, beta, theta, iota, omicron. This is a bacteria that's extraordinarily good at eating polysaccharides. And we profiled all roughly 100 polysaccharide utilization genes in this bacteria in mice monocolonized for that. What you see here on the left are 2 different images taken from the same cross-section of the mouse colon. And the green and red dots represent the distribution of individual RNAs imaged in beta theta. What I want you to notice is that the green RNAs are all enriched right at the mucus layer produced by the epithelial cells in the host, whereas the red RNAs are all found out in the lumen. And we see this across many, many, many different RNAs. And what's exciting is that the red RNAs all encode genes that utilize dietary polysaccharides, whereas the green encode genes that utilize the polysaccharides that we decorate our mucus with. And so we can see that beta theta fine-tunes its gene expression on the micron scale based on polysaccharide availability in the gut. Of course, this is a very simplistic proof of principle, but I hope you can see how this type of measurement can now allow us to characterize the full complexity of gene expression in complex microbial communities. So we come back to this image that's an inspiration to us. And I hope I've been able to convince you that MERFISH allows us to reveal some of the hidden complexity here. We can define cell types and states on the host side. We can chart how they remodel both molecularly and spatially when we have inflammation, and we can do similar style measurements on the microbial side. So if you are left with anything, I want you to be left with an understanding of what is MERFISH. MERFISH is really nothing more than multiplex single molecule fish, where instead of performing one single molecule fish image, we perform a series of those images, creating a stack of fluorescent on-off patterns for each molecule that we target, with each molecule discriminated by a specific optical barcode. What we've shown now is that this very straightforward idea can be used to massively multiplex these measurements. We can target hundreds, thousands, or tens of thousands of RNAs. By leveraging these error-robust and correct encoding schemes, we can do so with very high performance. And one aspect that we're very excited about that I have not highlighted perhaps specifically is imaging-based methods can be very, very high throughput. We can quantify tens of millions of cells and hundreds of thousands, tens of millions of RNAs and hundreds of thousands of cells. And it's routine for us to collect multi-million cell data sets, all for about a tenth of a penny per cell. So what is MERFISH? It's a tool for genomic-scale microscopy. And what I hope I've been able to do is to convey to you some of the excitement we have, because we think this is the tool that now allows us to not only define the parts of these complex systems, but how those parts fit together. So with that, thank you so much for your time and attention. And I want to thank all of the very talented people in our young lab that made contributions here: Yanyu and Paulo, Rosalind, Evan, and Ari. As I mentioned, many of these projects were collaborations with some remarkably talented labs here and labs that have since moved elsewhere. Peter Karchenko's lab and Ronnie Narwarski's are only just 2 of those. And if you're interested in learning more about MERFISH, I'm very happy to answer any questions you may have or follow up offline. So thank you.
That was a very exciting talk. Maybe a couple questions?
Like 3D rotating thing of an E. coli cell? Are you actually measuring these in 3D?
Yes.
Here in the online.
Yes, so I'll repeat the question. The question was, we had a very artistic rendition of an E. coli spinning around, And that suggests that these molecules are actually resolved in 3 dimensions. And the answer is we do that. So we will create Z stacks. We'll stack optically through every one of our samples. And that gives us basically X, Y, and Z resolution for each of the molecules. Please.
And they're very interesting, but they're very spread out when you do gut imaging. I would say, like, how would you do MERFISH to actually characterize the subtypes of enteric neurons?
Yeah, it's an excellent question. So to repeat it, the question was focused on enteric neurons. We have some in the data that I showed here, but with the preparation that we do, we capture very few numbers of enteric neurons per slice. So we have a second study where we cross-section the gut exactly as I've shown here. That's characterized pretty much in ileum all the way down. It's about 2.2 million cells. And even though we do nothing to enrich for enteric neurons, we have something like 30 or 40,000 enteric neurons that we capture in that data set. And so that's one of the hidden advantages of very, very high throughput imaging, is that these rare populations can still occur in very high frequencies. And we were very pleased. We just co-integrated this with some single cell data from a very specific study of the ENS. And we recapitulate all the cell populations and perhaps show some additional subdivisions. So very exciting. Great question. Yeah. Yeah, it's a great question. And so the question was to ask, how well do these type of image-based approaches work for cells of complex morphology? The actual identification of RNAs doesn't care about morphology at all, so that's not a problem. But cell segmentation, of course, does. And so I think, I'll say 2 things. Cell segmentation is not at all a solved problem. In fact, that term is an umbrella term to capture perhaps a huge diversity of computational challenges. And not just computational challenges, but experimental challenges. For example, if you want to chart the cellular RNA diversity in neurons, you probably want to have markers that fill the axonal or dendritic processes. Right? So I think it's not just that you need good algorithms for segmentation, but you also need to match the experimental work to that as well. But we see that, for example, in work that we piloted MERFISH in the brain, we capture the soma of cells, but we don't trace neuronal processes. But we see all these RNAs outside of the cells, extrasomal. Those are enriched for all of these classically transported, axonally transported RNAs. We actually see the same thing in the gut as well. We can capture RNAs that are clearly from the enteric nervous system out in the lamina propria, presumably because there's a small projection into that lamina propria that we're not defining optically. So I think this is doable, but depending on how you stain your sample and depending on how you segment, you just have to be cognizant of the things that you will properly segment and the things that you won't.
Great. I'm going to take a question from online. You can read it too if you want. From Theodore Tyler, what are the arrows based on the plot showing polarized cells and inflammation? Are they based on a certain analysis, or is the polarization visually determined based on the expression of inflammatory genes?
That's an excellent question. So that on that slide where I just very briefly summarized a tremendous amount of biology in terms of the way in which cells polarize, the arrows are my artistic rendition of polarization. No sophisticated computation. So the identification of those populations and the linking of what we think are the trajectories of polarization, those are done via standard clustering methods and pseudotime analysis methods.
Here's one from over here. Questions? Oh, I saw one. All right.
Please.
I imagine that you can't really do serial sections because things are going to change too much to match. But could you maybe do a 2-stage analysis where you select your 1,000 genes, find which of the genes that are the key ones for your pattern, then you do a second set where now you do for all the other remaining genes, let's say 100 genes were the key ones for your pattern, so you do as many sections you need with like 900 random genes. So then see if you're actually missing anything from transcriptome versus the selection of the 1,000.
Yeah, let me try to see if I can recast the question. Maybe stand in front of the mic while I do it. So it's a very sophisticated multi-part question. So the first thing is to ask, can we do serial sections? And I think perhaps I misspoke. We can absolutely do serial sections. We can take one slice to MERFISH, take the next slice to MERFISH. That's doable. We don't often do that because we want to try and get statistically distinct samplings of the spatial organization of tissue. The second question is to ask, I think the thread you're pulling on is a very good one. is, are there ways for us to do iterative targeting of different subsets of genes and perhaps expand the biology we can learn? And I think the answer is absolutely. There's some really interesting computational work looking on how one could potentially take transcriptome-wide data, like single-cell RNA sequencing, and find a reduced kind of dimensionality of that data that you could then measure with MERFISH that could then best predict what you see there. I think this is a fruitful line, and you could do it in an iterative way, like you suggested. But we actually we slightly shift the type of questions we ask with these data so that perhaps we're not so worried about capturing transcriptome-wide. We want to be able to define cell populations, but when you have 1,000 genes, that's typically not a problem. So most of the markers are there just to label these clusters. Then what we really get interested in is we take comprehensive coverage of these specific pathways. So now we're really doing a MERFISH atlas to understand cytokine and cytokine communication. If we've missed important adhesion molecules, it's okay, that wasn't the question that we were asking. So that like subtle shift in how we ask questions of this technique, I think, allows us to sidestep potentially that challenge of not having transcriptome-wide information.
I'm gonna take the last question. My privilege. So you talked about these very big sizes of data sets, for example, the 52 slices, which were 50 terabytes. Can you talk about the file formats that you envision researchers being able to share data in a meaningful way, and strategies that maybe we can use to compress data and make it series? Okay.
Yeah, great question. All of that. So, yeah, I think we ourselves go into a lot on file formats. I've been following the next generation imaging file format discussion with a lot of interest. Everything we do now is TIFF. I think we'll likely move everything to HDF5. So, I think I worry less about file formats, though I'd be happy to talk at length with anyone about this. We worry a lot about distribution of these data and staging of these data and storage of these data because, we build our own microscopes so that we can image on the scale that we want to. And with off-the-shelf components right now, we could be imaging 100 times faster. So like the scale of data that we could collect is just monumental. And one of the scary things is that the biology actually demands it. There are spatial structures we see that we're underpowered in, especially in recovery. And I don't know how you distribute petabytes of data. I don't think we have any way to do that. I don't think we have budgets that can support Amazon supporting these things. I don't think we have the ability to pay reproducibly for the compute of these things. And so I think we as a community have to grapple with this notion that some elements of these data are very difficult to think about how you stage and distribute. One way that I think we absolutely solve the raw data storage and analysis problem, which I think really provokes strong emotions when I say it, is that I think we embrace lossy compression. I think that's the short answer. Yeah, I told you, strong emotions. But you can. Every time I snap an image of the same sample, Poisson noise pixel values will not be the same. So if I can compress in a way that when I decompress, it's within error of Poisson noise, how is it any different than if I just snap that image a second time? It isn't. So that's where I think with the discussion goes. All right, I said
we're going to leave that on that wonderful note. Come talk to Jeff. We're going to transition into the next round. Thank you, Jeff. Wonderful presentation.